as we talk about all these new advancements such as IoT, RMM, cytometry, what do you see as the key challenges in implementation of these technologies, especially within the pharma industry? And I'd like to focus more on the rapid microbiological methods.
Okay, we know we used to operate TOC measurements and conductivity measurements and have an online control about the situation in the systems. And the only last or relevant time-consuming test method is the plate count bacteria tests, which have to be done in the laboratory, and the result is given 5 days later. So when you have a microbiological problems, you know it 5 days later. So the next step with regard to full picture of a water treatment system is an online bacteria counter. And there are some developed at the moment, and this is currently discussed how to implement this to replace also the plate count methods for the release of the product. Even this is the final goal, that we don't want to take samples and gain the bacteria, grow the bacteria on plates. And therefore, this is the final goal. But we are in a very early step of this development phase. So we are currently working to get validated systems and regulatory approval, so that we ensure that we meet the stringent regulatory requirements and gain acceptance from the authorities. Current problem is that we have no method standardization with regard to the online bacteria counter. So we have different technologies to evaluate these microbiological water quality. There's the flow cytometry, laser-induced fluorescence, enzymatic activity, and ATP, 4 different methods with pros and cons for all of them, and different units. It's not a count per ml as we used to have it in the past. We have the limit value, and then we have 50, 60, or I don't know, better 5 CFUs per ml. And this is one of the issues we have at the moment. And of course, these devices are not for nothing. They have relatively high costs for the initial investment. You have to calculate with about $100,000 plus to install such a system. And there are also consumables, especially, for example, in the flow cytometry, you need some colored solutions to color the bacteria before you count them. And there are also running costs, but finally, I think under the bottom line, we can save a lot of money when we get rid of the laboratory methods. That's one topic. And the other reason for this technology is that we can gain a full picture of the situation. So we can measure downstream reverse osmosis. So we can identify immediately a change in the microbiological water quality and can act immediately while doing a hot water sanitization or something else. So this is another big advantage. And another reason or problem is that we find much more bacteria than before on the plate method. So because we count also the viable but non-culturable bacteria. So not every bacteria in the water is able to grow and to grow on a plate. It's only a small part able to create a colony on a plate count. And so therefore we measure 5 to 10 times more bacteria than before. So we have to find new limit values, and this is a big problem. And depending on the measuring method, on the other hand. So and then maybe the last challenge, but this is specific to some methods, that we have the risk of false positives due to particles. When they have their self-fluorescence is on the same level in the bacteria, we may count particles as a bacteria. So for example, when you have PTFE particles from seals or ruching in the system, there is a risk of false positive. So there are a lot of challenges, as you can see, but nevertheless, people try to evaluate these technologies, have test plans, and so we will. The pharmaceutical industry is slow and conservative. It will take time, but it's on the way.